Hi Year 12s. This is just a short video to look at topic 1.7, which is biotechnology tools and techniques. In this video, we're just going to look at the biotechnology tools. So to start off with, these are the SACE key ideas, and as always, this is what I encourage you to be coming back to and using to be taking your notes and for studying. So you need to understand that DNA can be extracted from cells and that modern techniques can be used to analyze even small amounts of DNA. Segments of DNA can be multiplied using the polymerase chain reaction PCR. And you might have heard of PCR to do with COVID tests. So COVID tests, the ones where you have to go and get the nasal swab, are called PCR tests. And so we're going to look at what that actually means and what they actually do in that process. The base sequence of DNA can be determined by gel electrophoresis. So we're going to look at how to make a DNA profile and a DNA fingerprint. We're also going to look at something called electropherograms, which are types of graphs that illustrate DNA sequences. And lastly, we're going to discuss the ethical, economic, and cultural issues related to the collection of genetic information. So, as I said in this short video, we're just going to look at tools. So genetic engineering is about changing the genetic structure of organisms, and there are specific biological tools that are needed. These are listed here, but I think we're just going to get straight into them. So the first one are restriction enzymes. So restriction enzymes exist naturally in bacteria, and bacteria contain a lot of different restriction enzymes, and restriction enzymes cut DNA. So bacteria use restriction enzymes to cut up the DNA of invading viruses in order to kill the virus. So geneticists and biologists can use restriction enzymes to cut DNA at specific sites. And so there are thousands of restriction enzymes, and each one cuts DNA at a particular base code, and this is called a restriction site. So in this image here, you can see that this restriction enzyme makes a cut like this in the double strand of DNA, and it makes what is called sticky ends. So if we look on the next slide, some restriction enzymes will make that kind of cut like this, which gives us sticky ends. Some will just go straight down, and that gives us blunt ends. Which we're going to look at in a moment, if we have sticky ends, that's ideal for then binding to another sequence of DNA because it makes sure that it's going in at the precise position in the DNA strand. So the next tool is a gene probe. So if you think about DNA, you've got heaps and heaps of DNA, and it's really tiny, and we're trying to find a really specific section. We're trying to find a specific gene. So what we can use is a probe. So a probe is a short sequence of RNA or single-stranded DNA, and it has complementary bases to a section of the target gene. So if we heat up the target DNA, the strands will separate, and then so the strands separate, and then we can add in these probes, and the probe will bind at a specific site because of those complementary bases. Something else that's really useful is that probes are often radioactive or have a fluorescent marker, which then enables them to be located. So probes are used to find and locate a specific section in a target gene. The next tool is a plasmid. So again, plasmids are found in bacteria. Plasmids are circular DNA molecules, okay, and they often contain antibiotic-resistant genes. So when we think of a bacteria that is resistant to antibiotics, that's because of a gene that is found in a plasmid in that bacteria. So what we can do with plasmids, what geneticists do, is they can cut the plasmid once using a restriction enzyme, and it will give them those sticky ends. And then if they've also cut a target gene with the same restriction enzyme, it's got those sticky ends that are complementary, and so it can be inserted into the plasmid and it will bind together to then make a plasmid with the gene inserted. So this diagram probably makes that a bit clearer. So A plasmid might also be called a vector. Restriction enzymes will cut the DNA sequence of interest as well as the plasmid. So we use the same restriction enzyme, and then we've got the sticky ends, which are complementary so that the DNA of interest can be joined into that plasmid. And there's another enzyme called DNA ligase, which is kind of the glue. So restriction enzymes are the scissors that cut the DNA, and then DNA ligase will join those DNA sequences together. And then we end up with a plasmid that has the gene of interest or the DNA of interest within that plasmid. What we can then do is put this plasmid back into bacteria, and when the bacteria undergoes protein synthesis, that gene will be, like the protein will be made, and so we can have a desired result. And one application of this is people that with type 1 diabetes, they need to inject themselves with insulin because their body's not making insulin. And so that's made by putting that gene that codes to make for insulin into a plasmid, into bacteria, and then the bacteria will make the insulin. Okay, we just spoke about ligase enzymes, so this is another tool. So ligase enzymes glue DNA fragments, the sticky ends, back together. So they might glue, for example, the desired gene and the plasmid back together. When we've got DNA joined together from different sources, that's called recombinant DNA. So a plasmid that has a gene of interest inserted is referred to as a recombinant plasmid. So here's another diagram showing that if we've got a plasmid, double-stranded DNA here from a plasmid, the restriction enzyme will make a sticky end there, and it will do the same in human DNA, and then DNA ligase can be used to join them together to make that recombinant DNA. Here is another diagram of that one. Okay, the next tool, which we're going to look at more in the next video as well, is called gel electrophoresis. So electrophoresis is a tool, and it uses this device which has some gel and an electric charge running through. So electrophoresis separates and sorts DNA fragments according to their length. So if we have a DNA sample, if we cut it at specific sites, and then everyone, say my sample of DNA and your sample of DNA, if we cut them with the same enzymes, it's going to produce DNA of different lengths. So then what we can do is put the DNA into what's called a well in the gel. And because DNA has a negative charge, when we start that electricity running through here, DNA wants to move towards the positive electrode. And so what happens is the DNA that is smaller will move quicker and faster because of that negative charge towards the positive electrode, and the heavier, longer strands of DNA won't travel as far. So it's called running a gel. So you put the electricity through the gel for a certain amount of time, and then you stop it, and the DNA can be identified, and its length can be identified according to how far it's traveled through. And then what this makes is, so this is called a sample ladder of DNA fragments, and it could also create an electropherogram. So here's just another diagram of gel electrophoresis. DNA samples are loaded into the wells at the negative electrode. The power is turned on, DNA fragments migrate through the gel. The fragments are separated by size. So what's happening in these columns is that that's 4 different samples of DNA. And the power is turned on, then it's turned off, and this is what we end up with. So different patterns, which can be used to make a DNA profile of someone's DNA. Again, just comparing DNA fragments, this would be from 5 DNA samples. And so you can see that everyone has a different pattern in the electropherogram. Another tool are primers. So a primer is a short single-stranded sequence of nucleic acid, shown in this diagram in the red, and it provides a starting point for artificial DNA synthesis. So this is important for PCR, polymerase chain reaction to make multiple copies of DNA. So if we have the template DNA, and if we use heat to separate the strands of the template DNA, the primers will bind to the template, and then an enzyme called Taq polymerase will go along and synthesize the rest of the chain. So it's important that the primer has complementary bases to bind in the correct spot, and then there's free nucleotides which Taq polymerase will add on to replicate this DNA. And there's just another diagram that might help you understand that. Okay, so that's the end of our tools section. You have to go to the next video to look at techniques.